Stem cells are used in multiple pathways to achieve RGCs protection, such as paracrine function, working in multiple pathways to protect the RGCs and preserve its function. However, the sequence of events in axonal degeneration differs from that of somatic tissues, and therefore, strategies to protect axons may differ from those used to protect the cytosol. Neuroprotective approaches aimed at protecting the neuronal cell body may not be sufficient to improve function if the axon remains damaged and disconnected from its central nervous system targets. Stem cell research has not yet resolved the complex afferent retinal connections and long axons of RGCs for precise projection to the brain. Several neuroprotective strategies have been studied, such as peripheral nerve grafts, electrical stimulation, and the application of NTFs, including brain-derived neurotrophic factor, BDNF, ciliary neurotrophic factor, CNTF, glial cell-derived neurotrophic factor, GDNF, and nerve growth factor, NGF. Additionally, direct endogenous regenerative stimulation, RNA interference, and human adult stem cells have also been investigated. Stem cell-based therapies can aid in retinal regeneration by addressing two primary issues. 1. Preventing secondary degeneration of RGCs and preserving residual vision. 2. Replacing degenerated RGCs and promoting regeneration of RGC axons in the damaged area. The initial approach, known as neuroprotective therapy, involves incorporating stem cells into the deteriorating retina to create a nourishing environment for damaged RGCs. This can result in both anatomical and functional improvements. The second approach, known as RGCs replacement therapy, aims to replace damaged RGCs with healthy RGCs, or RGC precursors. However, the sequence of events in axonal degeneration differs from that of somatic tissues, and therefore, strategies for protecting axons may differ from those used to protect the cytosol. Neuroprotective methods alone may not suffice to improve function if the axon remains damaged and disconnected from its central nervous system, CNS, targets, despite the protection of the neuronal cell body. It is important to note that different RGC subtypes exhibit varying axonal regenerative capacities. Axonal regeneration is a complex process that involves many intrinsic factors and extrinsic environments, such as signaling pathways, epigenetic modifications, and inflammatory stimuli. It is suggested that no single factor is sufficient to promote regeneration. Identifying additional factors that regulate axonal regeneration will facilitate a better understanding of the underlying mechanisms. Therefore, many questions still need to be answered regarding the primary goal of axonal regeneration, which is to restore functional visual ability. Although regenerated RGC axons may be present at the target site in the brain, it does not necessarily imply the restoration of visual function.